This is Dr. Mitch Robinson, and this video is on insulin and glucagon, and it accompanies handout number 14 for cellular and molecular medicine. In this video, we'll examine the hormones insulin and glucagon. We'll look at the structure and the synthesis of these hormones and their effects on metabolism. The primary role of the pancreatic hormones is the regulation of whole body energy metabolism. These hormones regulate the concentration and activity of many enzymes involved in catabolic and anabolic pathways. Now, the pancreas has both endocrine and exocrine function. And the endocrine function is the production and release of the hormones that regulate metabolism. We're going to focus on two of these hormones, insulin and glucagon. The original evidence for the activity of insulin came from observations that removal of the pancreas from animals produced symptoms similar to those of diabetes in humans. Insulin was eventually isolated from pancreas and found to induce hypoglycemia. Insulin continues to be a major focus of medical research. It was the first protein to be sequenced and the first to be made using recombinant DNA technology. Insulin is a member of the insulin-like growth factor family. They're all structurally and functionally similar molecules. The tertiary structure of all of them is similar, and they all have growth-promoting activity, but the dominant role of insulin is metabolic. Now let's look at the structure of insulin. Insulin is a heterodimer. It consists of an A chain and a B chain, which are linked by disulfides. It's a fairly small molecule with peptide chains of 21 and 29 amino acids. The amino acid sequence is highly conserved among vertebrates. Insulin from one mammal is usually biologically active in another. Human insulin is very similar to bovine and porcine insulin. It only has one or two amino acids different. There's no appreciable difference in the biologic activity. Bovine and porcine insulin were standard therapy for diabetics for years. Now, virtually all administered insulin is human recombinant insulin. The figure on page 3 shows the amino acid sequence and the processing of insulin. Insulin is first synthesized in the endoplasmic reticulum as pre-proinsulin. The signal sequence is first removed from the amino terminus to form proinsulin. The signal sequence directs the molecule to processing that will lead to its packaging in secretory vesicles. The proinsulin molecule is then cleaved by a protease, and a section of the chain is removed that's known as the C-peptide. This leaves two peptide segments, the A and B chains, that are linked by disulfides. The insulin molecule and the C-peptide remain in vesicles near the plasma membrane, and on stimulation, these granules fuse with the plasma membrane, and insulin and the C-peptide are released into the blood. This is a molecular modeling representation of the insulin structure, and it shows the A and B chains in different colors. And this is a space-filling model, gives you a better idea what the actual shape of the molecule is. Again, the A and B chains are in different colors. Now, insulin forms dimers in solution, and then 3 of these dimers form a hexamer around a zinc molecule. And in this molecular model, you can see the hexamer, and this is 2 dimers, 2 insulin molecules. Each insulin molecule, of course, has 2 chains, the A and B chain. And this is a space-filling structure, closer to the true shape of the insulin hexamer. As I mentioned, all insulin is now made using genetic recombinant technology, and this allows us to make very specific changes to the amino acid sequence. So we can make designer insulin molecules that can be mass-produced from genetically engineered bacteria or yeast. So let's look at some of the insulin analogs and see how the molecular changes affect the biologic activity of the molecule. As I mentioned, insulin is stored as a hexamer, and these dissociate into monomers over time. Only the monomer binds to the insulin receptor and is active. The time course of insulin activity depends on how fast it dissociates into monomers. Zinc combinations of insulin form hexamers, and these are used for slow release. Analogs of insulin that don't form hexamers as well and dissociate faster are faster acting when injected. Eli Lilly and Company created the first insulin analog with Lispro. It's a fast-acting insulin analog. Its trademark as Humalog. It was engineered so that the penultimate lysine and proline residues on the C-terminal end of the B chain are reversed. Now, this modification does not alter receptor binding, but it blocks the formation of insulin dimers and hexamers. So this is a much faster-acting insulin. Another fast-acting insulin is insulin aspart. The amino acid B28, which is normally proline, is substituted with an aspartic acid residue. This creates a charge repulsion and prevents the formation of hexamers. Again, this creates a faster-acting type of insulin. Glulisine is a newer, rapid-acting insulin analog. The amino acid asparagine at position B3 is replaced by lysine, and the lysine at B29 is replaced by glutamic acid. Again, the changes result in non-hexameric, faster-acting insulin. Other insulin analogs have delayed action. Glargine insulin was developed by modifying amino acids, including the two positively charged arginines added to the C-terminus of the B chain. And this shifts the isoelectric point from 5.4 to 6.7. And this makes glargine less soluble at physiological pH. So when glargine is injected subcutaneously at pH 4, most of it exists as an insoluble precipitate. Now the insulin molecules slowly dissociate from the precipitate, and so they become available over an extended period of time. Dedimer insulin has a fatty acid, a myristate or 14-carbon fatty acid, attached to the lysine at position B29. This fatty acid causes the analog to bind to albumin in the blood, and this slows dissociation from albumin, allows it to act over a long period of time. Now, it's not necessary for you to commit all these analogs to memory. What's important is that you know the molecular basis of the fast and slow-acting analogs. Now, let's look at the release of insulin from B cells. Although the level of glucose in the blood is the most significant factor affecting insulin release, there are other agents that can elicit a response. The digestion of food causes the release of gastrointestinal hormones into the blood that stimulate insulin secretion. Other hormones, such as epinephrine, inhibit the release of insulin. A number of drugs have been developed that can stimulate insulin production. These include sulfonylureas. Fructose, unlike glucose, does not stimulate insulin secretion from pancreatic beta cells. The lack of stimulation by fructose is likely due to the low concentration of the fructose transporter GLUT5 in beta cells. The figure on page 5 shows the mechanism by which insulin causes the release of insulin granules from the B cells. Pancreatic B cells contain the GLUT2 transporter, which allows glucose to pass into the cell when glucose is elevated in the blood. Glucose is converted to glucose 6-phosphate by glucokinase and eventually results in ATP production. And ATP production in the B cell is linked to depolarization and an influx of ions. There's an ATP-gated potassium channel in the cell that's normally open, and it allows potassium out of the cell. An increase in ATP causes that channel to close. When the potassium movement stops, the cell becomes depolarized, and this allows calcium into the cell. Now, calcium is also released from intracellular stores. Calcium release leads to fusion of the granules with the cell membrane and secretion of insulin into the blood. So the beta cell acts as a glucose sensor, and it releases insulin in proportion to the level of glucose in the bloodstream. The sulfonylurea drugs are used for treatment of diabetes because they promote closing of the ATP-gated potassium channel. So these drugs make it easier for the cell to close the channel and depolarize and release insulin. Now we can examine the metabolic effects of insulin. Insulin has both growth-promoting and metabolic effects. The growth-promoting effects are slower and require a larger amount of hormone, while the metabolic effects are faster and require a smaller amount. Looking at the metabolic effects, insulin stimulates an increase in the transport of glucose into cells. In tissues such as adipose and muscle, this is due to the insulin-sensitive GLUT4 transporter. Insulin also stimulates the transport of amino acids into the cell. It stimulates potassium uptake and sodium efflux out of the cell. As we've seen, insulin has a pronounced effect on metabolic pathways. It increases glycogen synthesis in liver and muscle. It increases the rate of glycolysis in the liver, resulting in the conversion of glucose to acetyl-CoA. It increases fatty acid synthesis in the liver. And it increases triacylglycerol synthesis in adipose tissue and liver. Insulin inhibits the complementary pathways. It decreases glycogen breakdown in liver and muscle. It decreases fatty acid oxidation. It decreases triacylglycerol hydrolysis, and it decreases gluconeogenesis. As I mentioned, insulin also has growth-promoting effects. This includes an increase in DNA synthesis and an increase in cell growth and division. In adipose tissue, insulin leads to fatty acid synthesis and uptake and the production of triglycerides. It increases the activity of the pentose phosphate pathway to supply NADPH for fatty acid biosynthesis. In the liver, insulin increases the uptake of glucose, but this is due to the activation of the pathways that utilize glucose. It doesn't directly affect the glucose transport. Insulin increases glycogen synthesis by decreasing cyclic AMP and activating the protein phosphatases. Several regulatory enzymes become dephosphorylated. Insulin stimulates the activity of the pentose phosphate pathway. As we've seen, insulin stimulates glycolysis and inhibits gluconeogenesis. Carbohydrates are converted to acetyl-CoA. Fatty acid synthesis increases, as does the release of lipoproteins. Now, glucose transport into the brain is not dependent on insulin. Insulin does, however, act as a signal to reduce hunger through the receptors in hypothalamus. Glucagon is a 29 amino acid peptide hormone that's produced and released from the A cells of the islets of Langerhans of the pancreas. It's composed of one peptide chain, unlike insulin. Glucagon is generally viewed as a hormone that opposes the action of insulin. It's secreted when the blood glucose is low, and it serves to protect tissues against low blood glucose. Again, the primary signal for the release of glucagon from the A cells is low blood glucose. Glucagon's release is inhibited by hyperglycemia and insulin. Glucagon stimulates an increase in glucose in the blood primarily by its effect on the liver, where the insulin-glucagon ratio determines the intricate control of gluconeogenesis and glycolysis. Glucagon stimulates glucose release of the liver by stimulating glycogen breakdown and by stimulating gluconeogenesis. Glucagon inhibits uptake of glucose by the liver by inhibiting glycogen synthesis and by inhibiting glycolysis. Now, glucagon binds to a specific receptor that spans the plasma membrane, and it acts through the intracellular second messenger, cyclic AMP. The figure on page 8 shows the steps leading to glucagon response. This will be described later by Dr. Tepke. The figures on page 8 compare the factors that cause release of insulin and glucagon. Insulin release from the B cells is stimulated by amino acids and glucose and inhibited by epinephrine. Glucagon release from A cells is stimulated by amino acids and epinephrine and inhibited by glucose. Notice that amino acids stimulate insulin release, and they also stimulate glucagon release. This effect prevents hypoglycemia after eating a meal that has predominantly protein. If you're fasting and you have low blood glucose and you eat a protein meal, this is going to cause insulin release, but there's no carbohydrate, no glucose entering the bloodstream. Insulin release with no carbohydrate could cause hypoglycemia. The release of both hormones in response to amino acids assures that there will be some glucose available, produced by the A cells, and this will maintain glycemia. The glucagon gene gives rise to a protein, proglucagon, in a similar way to the synthesis of proinsulin. Interestingly, tissue-specific processing of proglucagon gives rise to different hormones in different tissues. In the A cells of the pancreas, the processing of proglucagon by the protease prohormone convertase 2 produces a product glucagon. Proglucagon is also produced in the gut. Here, a different protease, prohormone convertase 1, cuts the precursor protein at different places in the peptide chain, and this leads to the release of different peptides. This includes a peptide known as glucagon-like peptide 1, GLP-1. GLP-1 has a major effect of enhancing the release of insulin in response to glucose stimulus, and it suppresses the secretion of glucagon. The result is that GLP-1 has the effect of lowering blood glucose. The drug exenatide, also known as Byetta, is a GLP-1 agonist, so it helps GLP-1 stimulate insulin release. Exenatide is used as a treatment for type 2 diabetes. Amylin, or islet amyloid protein, is a 37 residue peptide hormone that's released from the pancreatic B cells at the same time as insulin. Amylin slows digestion and it slows the glucose rise after a meal. It also helps to lower hunger. Pramlintide, brand name Simlin, is a new drug that's a synthetic analog of amylin. And that concludes this video on insulin and glucagon.